Yeah, so actually what Nitin was mentioning about model-informed evidence. So just showing or showcasing what has been proposed by FDA so far for this model-informed evidence. So FDA is emphasizing using this MIE approach, and this MIE approach is leveraging this computational modeling and simulation, so that how this quantitative modeling and simulation will help us in extrapolating or predicting the data. So, and this ranges from different dosage forms. So if you see in the slide, they are talking about the oral dosage form. In this dosage form, you can use this in silico approaches. What FDA is proposing that we are encouraging using this in silico approaches for predicting or for justifying the dissolution limits as well, because this is a normal scenario in the organization, the pharmaceutical company, that a lot of out of specification, out of trend dissolution data is coming. So because of that, the batch needs to be discarded. So can we use in silico modeling to enhance this dissolution specification? So these approaches can be justified. FDA is also encouraging that if you have a model developed and validated, then we'll give you this relaxation in the dissolution limits. This is one of the approach. The other one, what they talk about is the particle size distribution. So instead of trying different particle size, going through a lot of clinical studies to find that, okay, whether this particle size is doing justice with the bioequivalence, what they said that you can do in silico modeling using different particle sizes of the API, and based on that, we can provide you a waiver as well. They are also encouraging for this alcohol dose dumping. Based on the alcohol dose dumping in vitro and in vivo data, you can justify your formulation with respect to the RLDs. So there are some other approaches they are also encouraging for the oral dosage form, which is also talking about getting a bio waiver for a BCS class 1 or BCS class 3 drug. So usually class 1 drug is usually we are getting the bio waiver, but for class 3, now FDA is saying that even for class 3 drugs, we are open to provide you a bio waiver if you have validated model or in vitro, in vivo correlation or justifying the use of excipients in the formulation, then we can provide you a bio waiver even for BCS class 3 drugs. And also supporting for this role of pH conditions in the stomach or in the fat or fasting condition, if we can justify that how this pH change is affecting our absorption. So these type of approaches FDA is encouraging for the solid oral dosage form, that if you have a model based on the in vitro data or the CMC properties of the formulation or the API, and the model is validated, we are encouraging and we are giving you the bio waivers or the relaxation in the formulation development or in the drug development program. So this was so far capturing about the oral dosage form. They are also into this non-oral, and for this non-oral, which is also categorized as the complex generics, because still a lot needs to be explored in the non-oral formulation. Especially they focused on this inhalation drugs and also for the transdermal. So what they're proposing is that if you have a method or an in vitro approach on which you have built a model talking about the deposition kinetics of the drug in the lungs or in the liver, then we can provide you a waiver, or we can also do this dose optimization for the different formulations what we have developed. So, and also for the transdermal, they are saying that if you have a IVPT, in vitro permeation test, if the IVPT is correlating with the in vivo outcome, then in those scenarios as well, bio-waiver can be granted, or the drug development program can be streamlined as well. So, so this is this what is about IVPT, in silico IVPT, that is in vitro permeation test. So actually, they are also encouraging the use of models for the different release kinetics as well. If you can approach them that, okay, we have used USP1, USP2 or USP5 for doing the dissolution studies, and based on that in vitro data, we have built the model. So FDA is saying that we can discuss these type of approaches to provide a waiver or a justification for moving forward as well. So in the non-orals, the most talked about is the LAI, which is categorized as long-acting injectables, where liposomes formulations are there, and also this microsphere formulation. And this microsphere formulation acts as a depot in the body. So a lot of companies are having trouble with the in vitro release, because these formulations are injected in the body, they are lying there for one month to 1 year. So getting the characterization done for these type of formulation is very lengthy, time consuming. So FDA is also encouraging that if you can set up a correlation with the in vitro and the in vivo, We can give you a waiver. We can also try to reduce the sample size, the subject size, the sampling time points, and all these things. So this model integrated evidence, again, I'm trying to emphasize is that you have a model which is justified based on the data what you have generated. Then the regulatory body, what Nitin was mentioning, FDA, is encouraging these type of approaches, that if you have a model based on the data which is validated, then we can talk about the drug development program and how to move further by striking out some of the studies or the required BE studies or clinical trials. So, and these are the other examples what FDA is suggesting about the clinical study design. So they mentioned that if you have conducted a BE study and if you have performed a population pharmacokinetics, then based on the population pharmacokinetics data, we can also justify it if some sampling time points has been missed or some volunteer has misbehaved. So these type of justifications has also been encouraged by FDA. If we have done a population pharmacokinetics modeling, a validated model, they need to have a validated model as well. So this is in the same line that the practical study considerations, which is talking about the BE assessment. So nowadays what happens that we prepare formulation A, B, C, D, but we don't know which formulation will be similar to the reference product. So what we can do, we can build the model for all these different formulations what we have done. And based on the model, we can select the formulation, that is the rank ordering of the formulation can be done to proceed for conducting a bio study or a clinical study, so that we don't have any surprises. So this approach is also entertained or accepted by FDA. And talking about this PK or bridging study, usually what happens in phase 1, phase 2, phase 3, formulations are similar. But once you go from phase 3 to commercials or launch, then the formulation are somehow modified. So at that stage and at that scale, it's hard to do another study. So this bridging study, what we call that, doing a bioequivalence justification based on modeling for a commercial batch or the phase 3 batches can be done, and this is also accepted by FDA. So these are the model integrated evidence what FDA is suggesting, that you come to us with a model which is validated, and we are there to assist you, help you, or accept if it is completely fine. So, and now coming to this point where what AI was mentioned, that in this case, this utilization of artificial intelligence, which has been repeated by Nirnit as well, that this is again a component of our computational modeling and simulation. So it can do wonders, no doubt, but it has to be coupled with this quantitative modeling and simulation, because once it is coupled, then we can do this extrapolation to any extent. With this modeling and simulation, what we are doing with the data, if it is coupled with AI, then the extrapolation can have many bounds. So this is what we are trying to emphasize, that AI will be solving a lot of problems in future for the drug development, but it has to be quantified to some extent. Otherwise, only dealing with this qualitative things will be talking or something which is virtual in the air. So we have to quantify it to get the most out of it. So this is what even FDA is proposing, that can we have this coupling of quantitative methodology or the modeling with the artificial intelligence, because what we are doing is quantitative, so we can put a number on it rather than only saying black or blue. We can say 1 or 2, so that is more meaningful to assign a number. And once we use a quantitative model, we can do that. So this is in summary this MIE, which came into picture by FDA last year. And the idea or the crux of this is that even someone who is not well versed or not sure that whether this approach will be accepted by the regulatory body or not. So in this case, what FDA or the regulatory body or even in silico minds is helping, that we will prepare a roadmap and then we approach the FDA saying that, okay, this is our line of attack. And based on our approach, FDA can also suggest something or give a green signal, okay, it looks good, you can proceed further. So these are the things which can be done. So the first thing is that we should have a line of attack for this modeling and simulation thing. So based on this, we have also tried to capture what FDA has been doing in the recent past, that for the case studies, what they have approved. So this is talking about the in silico bioequivalence study. What I'm just not going in detail, but this is a BCS class 1 drug. and in this case, the drug was amlodipine. And based on the in vitro data and in vivo data, a IVIVC model was developed and validated. And based on the validated model, the bio study has been striked out. Means the bio waiver was granted. There was no need to conduct a bio study for this drug. And this is a case study from FDA. They have done that. The organization name is not there, but they have given the bio waiver for BCS class 1. I emphasize again, there is no pilot, there is no pivotal. A complete bio waiver was granted for this drug. Moving to the next slide, this is again in silico physiological based biopharmaceutics modeling. So in this case, a drug was used, means the idea was that can this in silico physiological based biopharmaceutics modeling can help us in accelerating our development or drug development activities and also not compromising the efficacy and safety of the product. So this was used for a drug called lysinurate, which is a diuretic drug. And for that, the drug, and the idea was to show this slide here is that the whole development has been done based using in silico modeling. So right from the development starts, the screening, the prototype, and then the bio study, the in vitro data, all this model development has been done right from the beginning. That's why they called it the physiological based biopharmaceutics modeling. And based on this, the efficacy and safety was established for this diuretic drug. And based on the predicted safety and efficacy, further the validation was done, but the drug development timeline has decreased significantly because using this in silico approaches, a lot of preclinical studies have been striked out because the preclinical studies were not in place, and based on the model informed data or the in silico data, company has proceeded further, and based on that the development was done. Now coming to this slide, which is talking about in silico local acting drug in colon. So what we have talked about is the plasma concentration so far. So FDA is also emphasizing it's not only the plasma concentration which is in the plasma, but we are also trying to, they are also entertaining the drugs which is locally acting, but it is taken orally. So the classical case is mesalamine, which is used for anti-ulcer. And the effect is there in the colon. The drug has to be deposited in colon. So in this case, whatever drug has been absorbed is not having any value addition because the local action is there for the anti-ulcer. So in this type of approaches as well, FDA is suggesting that you can go ahead with this in silico modeling. In this case, they have done IVIVC by using different dissolution profiles at different pH condition for the product. And if you see, they have justified for different doses form as well, starting with suppository, intravenous, suspension, and the extended release capsule. So for all these different doses form as well, they have showed that if the modeling is justifying the in vivo outcome, then you don't need to perform this in vivo studies, and we can proceed further. So now talking about this again, in silico evaluation of pH on bioequivalence studies. So because we know that a lot of time the doctors prescribe your PPIs once you are taking an antibiotic or any drug. So how this PPI is affecting your drug absorption, whether this high pH condition or the low pH condition is increasing or retarding your absorption. So these type of release profile or the absorption profile can be simulated by using modeling. So you don't need to do particular study that, okay, the volunteer is given a drug with a pantoprazole or omeprazole to see how the elevated pH is affecting the absorption. So we know how this elevated pH is going to affect the in vitro scenario. Based on the in vitro scenario, we can develop a model, then build a model and correlate it with the in vivo. So with the in vivo scenario, and a mathematical equation, what we recently called is the IVIVC, can be used to predict the outcome. So what we are trying to say here is that we don't need to perform a bio study. This in silico modeling and simulation activities can be used to predict the bio outcome. And this prediction is accepted by FDA. That's what we are trying to emphasize, that whatever the prediction we are doing using modeling, then that prediction is accepted if the model what we have developed is validated. Whatever the equation we have given, a mathematical equation which is correlating with your in vivo scenario, if that is validated, then the prediction is accepted by the regulatory, not only FDA, but even by Europe, Australia, South Africa. We have success stories for different global regulatory bodies. So we are working, in Silico Minds is working for FDA, Europe, Australia, Canada, South Africa, Brazil. So all these global regulatory bodies are accepting in silico modeling. So I will just go through some of the case studies what we have done so far. So for our clients and customer. So this is just example what we have been doing so far in the last 1 decade with ISM in silico mines. So the biowaivers is one approach. So we are helping our clients to get complete biowaiver. They don't need to do a bio study. Also talking about the population pharmacokinetics or the virtual bioequivalence. Because usually this, you have to do a study in 50, 40, 500 volunteers. We can simulate that study for you. So that is called a virtual bioequivalence study. So, and also talking about PVPK study, whether the dose for the child or for the adult should be same or different. So we, instead of doing the study, because it's hard to find a child volunteer for our bio study. So in that scenario, this PVPK modeling comes into picture. We can predict the dose for the children by doing or simulating the study. So manufacturing process is one of the approach as well, where in silico modeling can be used. Here different methodologies are used. This is a DEM approach, discrete element methodology, which is used to predict the scale up and the manufacturability of a formulation. In silico drug device combination, so again mimicking how the device and drug together will behave, how much dose will be administered with one actuator, what is the particle size, what is the force which is going in. So all these things can be modeled. And again, talking about formulation optimization, which is rank ordering the formulation, 505B, 505B2 support, where we can design a new doses form altogether with a different dose. Means we can decrease the dose, we can make a tablet to a capsule, capsule to a suspension. All these changes can be modeled, and we don't need to do study at each and every step. So I'm just jumping through. Actually, these are the case studies, generic case studies, what in silico mines has delivered so far. And again, talking about the in silico bioequivalence. So actually, if you can, it can be correlated with the FDA case studies as well. The in silico thing, what FDA has done for amlodipine, we have also doing the same for our clients and customers. That we don't need to do a bio study. By using in silico modeling and simulation activities, we can strike out the B or bioequivalence study. We can directly get a waiver for that. Again, talking about bioequivalence for the different particle size. So this is a very classical example where we got a biowaiver for a company, a commercial was going on, and for that the API manufacturer has changed the particle size. So the sponsor was worried that the particle size has been changed. The regulatory body is asking to conduct a bio study, a clinical study, and because of that the commercial will be stopped, and there was a big loss. But by using modeling and simulation approaches, we have modeled the different particle size, how this different particle size will affect the absorption once it is going through the GIT. And this absorption similarity was shown with different particle size. So in this case, a biopredictive media was first screened. That biopredictive media was used to see the differences of particle size and how this differences is translating into the absorption. So all these things were taken into account, and the prediction was done. And based on the prediction, we concluded that with increased particle size, there is no change in the absorption. So by using this approach, we have taken a biowaiver for the revised particle size as well. So looks like that we are running short of time. I will just quickly go through the slides. I'm not talking about it. So this was talking about the relaxation in dissolution limits for the commercial batches using in silico modeling. And this is again talking about the long-acting injectables. So if you see in the graph, that is for 56 days or 42 days, we have to do a study. So we can simulate that 42-day studies in 4 days. So this is how we are adding value or decreasing the development timeline as well. So this is a transdermal patch. This is how the patch, which is not oral, how this patch is behaving. We can simulate the skin physiology as well. The dermis, epidermis, hair follicles, how this is helping in the penetration of the drug with respect to the reference and the test product. So I think this is the one which is talking about the API. Just touching it, that with the in silico approaches, we are helping our customers for this, predicting the route of synthesis, the retrosynthesis, the reaction pathways, the yield, the isomers, the polymorph. All these things can be done using in silico approaches, and for this we are using AIML. So so far it was computational modeling, but once we go into this AI domain or the discovery domain, then we are coupling our in silico modeling with artificial intelligence and then also with machine learning, because we have access to large databases which can provide rank ordering or the confidence what we are predicting is in line or not. So these slides are talking about AI coupled with ML as well. So, and this is the discovery slide, which is again talking about a lot of ML based and then also the AI. So drug repurposing, molecular docking, bioactivity predictions. So all this protein ligand binding. And this is done using a lot of machine learning approaches. And then if this machine learning approaches is trained by having a lot of more data and more data, what we call it the AI. So these drug discovery AI thing is more or less driven by using machine learning and then the AI. The earlier thing what we're talking about for the generic space was the computational modeling or the quantitative modeling. So I think these are the things what we want to talk about so far, and running short of time. So I would be happy to take any questions if there is there. Yeah.